Ever wonder how we can predict what the next generation might look like? Well, today we're going to unlock a really simple but incredibly powerful tool that does exactly that. So let's dive in and solve the puzzle of heredity using something called the Punnette square. I mean, it's a question we've all thought about, right? You look at your family, and maybe you and your sister both have your dad's eyes, but your brother totally has your mom's hair. How on earth does that happen? Well, that exact question, that's the whole basis of genetics, the science of inheritance. So here's our game plan. We'll start with the big puzzle itself. Then we'll learn the basic lingo of genetics. After that, we'll meet our amazing problem-solving tool, and then we're going to build one together, step by step, to see what it can really tell us.
For centuries, people knew that traits were passed down. That was obvious. But the how? That was a complete mystery. So this first piece of our puzzle is just getting a handle on the fundamental process at play here.
At its very core, it's this simple. Inheritance is just the process of passing characters or traits from one generation down to the next. You know, it's the reason an apple seed grows into an apple tree and not, say, an orange tree. It's the foundation of it all. Okay, so to really crack this puzzle, we need to learn the language. It's kind of like cooking, right? Genetics has its own special terms for all the ingredients and the instructions that make you, well, you. First up, we've got the gene. The easiest way to think about a gene is as a single instruction in the giant recipe book of life. So there's a gene for your eye color, a gene for your hair texture, and in the case of Gregor Mendel's famous pea plants, a gene for how tall the plant grows. Now, for any given gene, there can be different versions, kind of like different flavors, and these are called alleles. So for that stem height gene we just mentioned, one allele might say be tall, and we'll represent that with a capital T. Another allele might say be dwarf, and we'll use a lowercase t for that. So since you get one allele from each parent, you always end up with a pair. This specific combination of alleles, like two big T's, a big T and a little t, or two little t's, is your genotype. This is the actual genetic code, the recipe that's written in your DNA. The phenotype, on the other hand, is what you can actually see. It's the physical result, the expression of that genotype. So if the genotype is the recipe, the phenotype is the finished cake. In our plant example, that means the plant is either visibly tall or dwarf. Okay, so let's just make sure this is super clear, because it's absolutely crucial. The genotype is the internal genetic instruction, the recipe. The phenotype is the external, visible outcome, the cake. Understanding this difference is the key to, well, everything that's coming next. All right, we've got the language down. Now let's meet the tool that brings it all together. It's a simple little grid that lets us predict the future. The genetic future, that is.
This tool was developed by a British geneticist named Reginald C. Punnette, and the square that's named after him is basically a probability calculator. It gives us this beautiful, clear way to see all the potential outcomes when we cross two parents. So we get the what and the why. Now for the fun part, the how. We're actually going to build a Punnette square together, right now, step by step. And you'll see just how easy it really is. Here's our scenario. We're crossing two pea plants. Now, they both look tall, but they each secretly carry that hidden allele for dwarfism. That means their genotype is TT, a mix of one dominant and one recessive allele. We call that heterozygous. So, what happens when we cross two of these TT plants? It's just 4 simple steps. Seriously. First, we figure out the possible alleles each parent can pass down. Then, we draw a simple 4-box grid. We label the top and the side with those parental alleles. And finally, we just fill in the boxes. Let's do it. Okay, let's build this out. Since each parent is TT, they can pass on either a big T allele or a little t allele. So we put one parent's options across the top, big T, little t. Then we put the other parent's options down the side. Again, big T, little t. Now, we just fill it in by combining them. The big T from the top meets the big T from the side, that gives us TT. The little t from the top meets the big T from the side, that's TT. Over here, the big T from the top and the little t from the side make another TT. And finally, the little t from the top meets the little t from the side, and we get TT. And just like that, we have a complete map. These 4 squares represent every single possible genetic combination for the offspring of this particular cross. It's all laid out perfectly for us. We've built the map, so now it's time to read it. What do these combinations, the TT, the TT, and the TT, actually mean for the next generation of pea plants? Let's just literally count what's in the boxes. Okay, we have 1 TT square, we have 2 TT squares, and we have 1 TT square. That gives us our raw genotypic results. So when we look at the genetic makeup, the genotype, we see this really clear pattern. The ratio is 1 to 2 to 1. What that means is for every 4 offspring, on average, we can expect 1 with the TT genotype, 2 with the TT genotype, and 1 with the TT genotype. But wait, remember that distinction we made earlier? This is the genotype, the recipe. What about the phenotype, the thing we can actually see? Well, this is where dominance comes into play. That capital T for tallness, it masks the effect of the lowercase t for dwarfness. So let's think about it. The plant with the TT genotype, it's going to be tall. And because that big T is dominant, the 2 plants with the TT genotype will also be tall. Only the 1 plant with the small t genotype will actually be dwarf. So that means we have 3 tall plants for every 1 dwarf plant, a 3 to 1 ratio, or you could say 75% tall and 25% dwarf. And this right here, this is the real magic of the Punnett square. It lets us see both the hidden genetic code and the visible results. The underlying genotypic ratio is 1 to 2 to 1, but that produces a completely different observable phenotypic ratio of 3 to 1. How cool is that? So we've figured out how to predict one trait, which is amazing. But life is rarely that simple, is it? The cool thing is, the same principles we use today are the foundation for tracking multiple traits at once, like a plant's height and its seed color. And that just opens up a whole new world of genetic possibilities. What do you think that Punnett square would look like?